or are we really just the same? Different from, for example, rats or dogs or whatever. And because we're in a cafe, I had to put this one. So, next slide. So, I guess, yeah, I thought I had a slide here on biology. Maybe they don't. So anyways, there are lots of naturally occurring diseases in pets. And this is the cool thing is that every day arriving into our hospitals are disease processes that mirror what's seen in humans. And that's why we pay so much attention to the literature that's going on in human medicine. So heart diseases, but dogs, of course, don't get heart attacks. Autoimmune diseases, trauma, bacterial infections, diabetes, arthritis, cancer, stroke, epilepsy, neurodegenerative diseases, Alzheimer-like diseases, disc disease, obsessive compulsive disorder. You can see why we're interested in the dog. Next slide. And dogs get a large number, in fact, of all the mammals, domestic animals, they get the largest number of inherited diseases that make them, the dog as a breed, have the greatest potential to model human inherited diseases. Next slide. And what do we get out of studying our pets and new therapies in pets and biomarkers in pets is we get an understanding of the safety of new treatments, their toxicity, how they behave in the bloodstream or how they're disposed of, that's the pharmacology and pharmacokinetics, where the drugs or the cells go to, the distribution, the importance of discovering the right dose, and of course the activity, the efficacy of those new therapies. Next. So, what are stem cells and why are they important for the last part of this talk? Stem cells are cells that have the unique ability to divide many times and turn themselves into multiple cell types. A pluripotent stem cell is capable of basically, in the lab, being induced to turn into any cell of the body. A multipotent stem cell has more limited potential. That can turn into more than one cell type. They also are unique because they live in a niche. They live somewhere and they hide somewhere in the organ, somewhere where they won't be damaged. Generally speaking, they're hiding from changes in oxygen, they're hiding from changes in nutrition. So there are these niches where you find these stem cells. Next slide. Embryonic stem cells are obviously from an embryo, the inner cell mass of an embryo, and they can be used to actually remove and turned into organ cells and used to replace damaged neurons or retinal cells. These are the two clinical trials that are going on right now with embryonic derived cells. Retinal pigmented epithelium and spinal cord supportive cells. You can also turn your own mature skin cells into embryonic-like stem cells, and that's the process of inducing pluripotency. And these are called iPSC cells, induced pluripotent stem cells. So we can each have our own cell line that's pluripotent, theoretically. And those cells can be used to replace damaged cells when they're transplanted into an organism. And then the bulk of stem cell research is being done on cells that are multipotent, that come from yourself or from somebody else, that are really not used to replace damaged cells because they don't really stick or graft into the recipients. These are cells, with the exception of bone marrow cells, which do. Most adult cells can't find a place and stick and survive into the recipient. So they're actually used in many clinical trials now because of their attributes as stem cells to activate regeneration without replacing cells. Next slide. So we use, in our clinical trials, we use adult stem cells. They're called mesenchymal stem cells because they're from the mesenchymal layer, one of the 3 layers of the embryo. And the way they work, as I said, they don't replace cells. When they're injected into a patient, either into the bloodstream or directly into an area of interest, the cells actually release a whole lot of chemicals. And those chemicals include microRNAs and proteins. So we circle back to microRNAs. They also release all these vesicles, these blebs. They bleb off all these things that stem cells bleb off these membrane-bound vesicles called extracellular vesicles that are picked up by recipient cells. Those vesicles, we're particularly interested in these vesicles. This is a high magnification electron microscope of these vesicles. They contain a lot of microRNAs and proteins that have tissue repair and regenerative properties. So we use the stem cells to cultivate vesicles, and the plan is to isolate these vesicles and use them therapeutically. And that's a huge movement in the life sciences right now. Next slide. There's a lot of resources that are involved in isolating stem cells and characterizing them and characterizing nanoparticle size extracellular vesicles. And so this is kind of what our lab looks like, except it's not divided up on a slide on a piece of paper like that. It's all in 3 rooms. The source of stem cells we're actually using in our clinical trial, we get from the umbilical cord. So why do we use the umbilical cord? So we have high volume practices are sending us sterilely removed umbilical cords from elective C-sections. The dogs are getting elective C-sections, they give us the whole placenta. Sarah usually is blessed with the task with the removal of the umbilical cords and other parts of the placenta. So first of all, why do we use the placenta? Placenta is like probably the most fascinating area for regenerative medicine research right now. Because it's a fetal organ, and it's free, and it's abundant, and people are throwing them away, it seemed ridiculous to us, and based on the literature that's out there that shows that these are really have fetal stem cell-like properties, to throw these away. So we've designed a program to actually focus on using umbilical cord tissue, not umbilical cord blood, but umbilical cord tissue to get our stem cells. Next slide. They're amazing cells, and we know that they release a lot of microRNAs that have the right epigenetic effect in disease processes that we're interested in. For example, our approach is to take a disease like mitral valve disease. Now this is where the valve in the left, between the left atrium and the left ventricle, gets really lumpy bumpy. You can all see that. And then you can see that that valve isn't going to close very well, which means blood's going to go backwards instead of forwards to your brain and everywhere. It's going to go backwards, regurgitate. You hear people having mitral valve prolapse or mitral valve regurgitation. It's quite common. It's actually common in people in their 20s. And don't even know if that problem is going to become a serious problem or not at that time. That's one of the issues. So these valves become lumpy bumpy, and Vicki Yang in our lab has been studying exactly what microRNAs are disturbed in the tissues of those valves. And she found 2 in particular, we'll just call them 29 and 30. And what she's done is we've designed therapies based on our umbilical cord cells and the vesicles they're releasing, because they have a large amount of these. So that means we could potentially use stem cells and their vesicles to resupply the microRNAs that control the disease process. So a lot of disease processes involve the loss of microRNAs, because these microRNAs are usually involved in silencing genes, and when they don't silence the genes go crazy. In the case of the mitral valve, they go crazy making matrix proteins like collagen, and they just turn into a big scar. And that's the problem. But if you could bring these microRNAs back up to normal physiological levels by supplementing them, and in this case using actual stem cells to do that, or even gene therapy, you could potentially reverse the course, or at least stabilize the course. So most patients that get mitral valve disease do not end up with heart failure, but a certain percentage do. So if you could block that process of our patients going into heart failure. Next slide.